Chapter 3, Segment 3 starts out by talking about how synapses can either be excitatory or inhibitory. So, the membrane potential of the the postsynaptic cells is briefly altered by neurotransmitters being released into synapses. Postsynaptic potentials are brief changes in the membrane potential of the postsynaptic cell. These potentials determine whether the neuron will reach threshold and generate its own action potential or not, and it is initiated by stimulation at a synapse, spreading passively across the membrane. It is important to remember that excitatory and inhibitory neurons get their names because of their actions on postsynaptic neurons, not because of how they affect behavior. So an excitatory postsynaptic potential, known as an EPSP, depolarizes potential in the postsynaptic neuron. It is initiated by stimulation at a synapse, and it is caused by excitatory connections. Excitatory postsynaptic potentials increases the probability that the postsynaptic neuron will fire an action potential. Synaptic delay, which is the brief delay between an axon potential, arrives at the axon terminal and when the postsynaptic potential is created. This reflects the time needed for the neurotransmitter to be released and diffuse across the synapse. The postsynaptic depolarization begins about half a millisecond after the action potential arrives at the presynaptic terminal. The combined effect of multiple excitatory synapses is typically needed to elicit an action potential in the postsynaptic neuron. Now, an inhibitory postsynaptic potential, known as an IPSP, hyperpolarizes potential in this postsynaptic neuron, which is caused by inhibitory connections. IPSPs decrease the probability that the postsynaptic neuron will fire an action potential. It is important to remember that inhibition counteracts excitation. So, IPSPs result from the opening of channels that permit chloride ions into the cell. It does not have to do with potassium and sodium as much as the other ones talked about in previous segments. So, inhibition also plays a role in processing information, turning neural switches on and off. All neurons use the same kind of propagating signals, but the effect of the postsynaptic cells differs greatly depending on which signal was used. So basically, the neurotransmitter being released determines if a synapse excites or inhibits. The nervous system walks a fine line between overexcitation, which is seen when people have seizures, and underexcitation, which is seen in coma and death. Whether a neuron fires an action potential is decided by this balance between the number of excitatory and inhibitory signals that it receives. Complex behaviors requires neurons to both integrate and transform messages. This means that neurons perform information processing due to their synaptic inputs. A synaptic input is the way in which neurons integrate the postsynaptic potentials and the trigger mechanisms that determine whether a neuron will fire an action potential. Information processing is adding and subtracting myriad inputs it receives from other neurons. So postsynaptic potentials are caused by depolarizing or excitatory or hyperpolarizing, which is inhibitory, transmitter chemicals. EPSPs and IPSPs spread passively over the neuron, which means that their strength decreases over distance. An electrode detects integrated membrane potential at the axon hillock. In later sections and in talking about spatial summation and temporal summation, it is what you inject into the axon to get your desired effect or observe an effect. When inhibitory synapses are also active, the potentials that excite and inhibit partially cancel each other out. This is seen when the neuron subtracts IPSPs from EPSPs and no action potentials are fired. This can be seen in the top of figure 3.10 on page 77. So, spatial summation is a summation at the axon hillock of the postsynaptic potentials from across the cell body. Summation comes from different physical locations, as seen in 3, and then it is influenced by distance. Convergence of excitatory messages from many presynaptic neurons are required for a neuron to fire an action potential. In temporal summation, however, there's only one input source, and the closer in time they occur means the greater the action overlap and greater summation occurs. So it's important to remember that although axon potentials are all or none phenomenon, the effect on the postsynapse is determined by size. The membrane potential at the axon hillock reflects the moment-to-moment integration of all the neuron inputs, but the axon encodes it into an ongoing pattern of axon potentials. Dendrites in the cell body can also produce postsynaptic potentials. Dendrites increase the amount of information the neuron can take in. So, all other things being equal, the further out a dendron potential is produced, the less effect the potential should have at the axon hillock. This is because, as mentioned before, as distance occurs, the signal gets weaker and weaker. So, information arriving at various parts of the neuron is weighted in terms of the distance. Synapses that are distant from the axon hillock compensate by producing larger postsynaptic potentials, which makes them more comparable to neurosynapses. On the bottom of page 78, a brief table overgoes all those characteristics and the action potentials of the excitatory and inhibitory postsynaptic potentials. So, on the top of page 79, there's a sequence for synaptic transmission that takes place. So at first, the action potential is going to arrive at the axon terminal. The depolarization then opens voltage-gated calcium channels, allowing calcium ions to enter. So calcium ions cause synaptic vesicles filled with neurotransmitters fuse with the presynaptic membrane, which releases neurotransmitters into the synaptic cleft. This promotes exocytosis. Exocytosis is the fusion of vesicles with the presynaptic membrane. Transmitter molecules cross the cleft and bind to receptor molecules in the postsynaptic membrane, causing ion channels to open. Ion flow creates either EPSP or IPSPs in the postsynaptic neuron. So this basically just means that synaptic transmission is the transfer of information across the synapse. As mentioned with exocytosis, V snares and T snares mediate it. V snares are the anchors that aid vesicles in fusing their presynaptic membrane. T cells are your target synapses, or they bind V snares to dock vesicles. Synaptogem responds to calcium ions to trigger vesicular exocytosis. And the kiss and run effect, which is vesicles fused with a postsynaptic membrane long enough to spill neurotransmitters, then pinch off to return to the presynaptic terminal. This is just a variation of exocytosis that the book talks about. And then the book also mentions how botulinum toxin and tentinous toxin silence synapses, which disables exocytosis and synaptic transmission. So when talking about the action of a neurotransmitter on a receptor protein, we use a lock and key analogy. This shows that a molecule of the correct shape, or a ligand, fits into a receptor site and activates or blocks it. A receptor molecule is a protein that binds and reacts to molecules of a neurotransmitter or hormone. An example being acetylcholine, which functions as both an excitatory and inhibitory neurotransmitter depending on what synapse it attaches to. This analogy is strengthened by endogenous ligand, which are produced within the body, or hormones, and endogenous ligand. The exogenous ligand, which is outside the body. This originates outside the body, so it's drugs and toxins. This segment concludes by describing the difference between an agonist and an antagonist. An agonist binds to a receptor molecule and initiates a response like that of another molecule, which is seen in nicotine and muscarine. And then an antagonist interferes with or prevents the action of a neurotransmitter, which is seen in Curare and bunjaro toxin. And that's pretty much it for this segment.